Hi students, good evening. So I welcome you all for the biology detailed solutions, the mock test 4. Mock test 4, biology detailed solutions, part 2 discussion. So I welcome you all for the biology. Yes. So we'll start up the session now. A warm good evening, students. Yeah. So now we'll start with the question discussion. So previous session we discussed the last 45 questions. And now we are going to discuss the next 45 questions. Okay, so we'll start the discussion now. So here there is a match the column. Match the column. 1 minute. Okay. So here we are going to match column 1. Yeah. Match the column 1 and with the column 2, and you are going to choose the correct option. Okay. Phaeophyceae, Rhodophyceae, mosses, Pteridophytes. So here they have given Funaria, Polytrichum, Sphagnum, Equisetum, Coleotum, Pteris, Ectocarpus, Dictoeta, Laminaria, Polysymphonia, Porphyriae, Gracilaria. So now we are going to match exactly what is it with each of the column. So now we have to know that first you should understand, okay, the basic that what you call the like how are we going to make it up with it. So when you look into it, like first when you see the Ectocarpus, Dictoeta, Laminaria, actually they are the members of the Phaeophyceae. Like what is Phaeophyceae? That brown algae, right? So they are the one. What about the Rhodophyceae? The Rhodophyceae, you can see the Polysymphonia, Porphyria and Gracilaria. What about the mosses? We have the Funaria, Polytrichum and Sphagnum. Okay, and then Pteridophytes, we have the Equisetum, Coleotum and Pteris. So this is how this goes about. Yes, Sri Vatsan, very good, welcome. So we have what is the Ectocarpus, Dictoeta and Laminaria. See, they are the members of the Phaeophyceae or the brown algae, that is primarily a marine habit. What about the Polysymphonia or Porphyriae, Gracilaria or the members of this Rhodophyceae, they are the red algae. They are marine with higher concentration, like they will be more in the warm areas, right? Then we have those mosses, okay, we have that Funaria, Polytrichum and Sphagnum, okay, they are the mosses that will grow mostly in the shady hilly areas. And what about the Pteridophytes? Like we are very well aware of that Equisetum, Coleotum and all that, they are the first terrestrial plants, right? They have those vascular tissues in them, the xylem and the phloem. Okay, so the right option is the first option. Okay, in the TCA cycle, okay, the FADH2 is formed during the conversion of succinyl-CoA to succinate, the conversion of citrate to cis-alconite, conversion of succinate to fumarate, conversion of fumarate to malate. So where is this FADH2 formed? Now first you should understand the cycle here. We know that from the glycolysis pathway we have the pyruvate. From that there is a link reaction, right? We have the acetyl-CoA and then that is entering the citric acid cycle, the Krupp cycle. Now we know that these are all the 6 carbon compounds that is being formed, and then there is a 4 carbon compounds, all this. So the succinate, like you can see here, from succinate the conversion to the fumarate, okay, there is the FADH2 is getting formed, that is FADH is getting reduced to FADH2. So where is the right answer? Yes, the answer is conversion of succinate to fumarate, that is the succinate is a 4 carbon compound and then the fumarate is also a 4 carbon compound, and what is the enzyme that is needed? It is succinate dehydrogenase. So that is the one which is going to help in converting the succinate to fumarate. Okay, so this is the right option. Okay, so the answer is conversion of succinate to fumarate. Okay, so select the correct answer regarding the phase of meiosis and their respective events using the list given below. We have prophase 1, metaphase 2 and anaphase 1. So they were asking us giving the correct answer. So crossing over occurs, sister chromatids migrate to opposite pole, homologous line up at the equator plane. So they are asking us to find like all three are correct, or if both are correct and 3 is false, or 1 is correct, 2 and 3 are false, or 1 and 3 is correct and 2 is false. First you should understand the crossing over. What is crossing over? There is exchange of the genetic material. Acha? So there is going to be exchange of the genetic material which is taking place between the homologous chromosome. Okay, so particularly that is going to happen in the prophase 1 of the meiosis. Okay, and that we call that synapsis. So this is the right one. Okay, now what happens? Now the homologous chromosomes, okay, they will get paired up. They will be lining up at the equator during the metaphase 1. Like you can see that lining up of the chromosomes in the equatorial plane. So that is what is going to happen up here, but rather what they have given up, see, they are lining up at the equator plane. But that sister chromatid migrate to opposite pole is the wrong one. Okay, and what about the anaphase 1? The anaphase 1, the homologous chromosomes line up at the equator. No, here what is happening in anaphase 1? The sister chromatids, because of that pulling of that thing, what is going to happen? That spindle fiber, now what happened? They will be pushing to the, they will be migrating to the opposite poles during the anaphase 1. So all these options are wrong. So what should be the right one? Yes, the answer is 1 is correct, 2 and 3 are false, because in metaphase 2 the homologous chromosomes do line up, but what happens in anaphase? They will be pulled apart. So that has to happen. So first one is correct where the crossing over occurs, but the second and third are false. Okay. Bryophytes differ from pteridophytes, that is swimming anthrozoids and independent gametophyte, archegonia, lack of vascular tissue. So how are we going to differentiate this? So the bryophytes differ from pteridophytes like where all? So we have to know that bryophytes, those amphibians of the plant kingdom, so how are they going to get differ from the pteridophytes? Now we have to know this bryophytes, okay, they have three, they are the three division of this non-vascular land plants, like the mosses, liverworts, hornworts and all that. So they are capable of surviving even in the drier environment. But what about the pteridophytes? You see, no, they don't have flowers or seeds. Okay, they are called the cryptogams, and they will include those ferns, horsetail, that equistem which we saw in the first slide. So here they are considered as terrestrial plants because they have those vascular tissue, the presence of xylem and phloem. So pteridophytes. So how do they differ? So in bryophytes, those vascular tissues of xylem and phloem are absent. So water and nutrients, they enter the cell by cell diffusion. But what about in pteridophytes? We have the vascular tissue of xylem and phloem. Okay, but they don't have the true vessels and all. Even in phloem, they don't have companion cells or not. So that is where. So what should be the answer? Yes, the answer should be, yes, the answer should be lack of vascular tissue. Okay, so the answer should be lack of vascular tissue. So that should be the right answer. Okay, so yes, Srivatsa, I wanted you to participate and give the answers. Is excellent. So lack of vascular tissue. Okay, now we'll go to the next question. It's a very interesting question. For the enzyme enolase, the substrate is succinic acid, 2-PGA, PEP and fumaric acid. So you should know that for the enzyme, okay, for the enzyme enolase, acha, so what is the substrate? Now we know that during the process of glycolysis, okay, let me tell you. So during the process of glycolysis, what is happening? It is going to convert the glucose, okay, into the 3 carbon molecule pyruvate. Okay, pyruvate. Now what is happening during the process? This mainly is to produce the ATP molecule. So what is happening? The enolase is the enzyme that is used to convert the mainly the 2-PGA, that is 2-phosphoglycerate to phosphoenol pyruvate. 2-phosphoenol pyruvate, which is somewhere around the 9th reaction in the glycolysis. It is a reversible dehydration reaction also. So the right answer is what? It is 2-PGA. So 2-PGA in the presence of enzyme enolase, it is going to release a molecule of water and it is going to form the phosphoenol pyruvate. So you are able to follow. So that is the right answer. Which of the following statement is incorrect? Okay, which of the following statement is going to be incorrect? We will see what are the statements. So the statements here is around the shoot Abhyas, those modify themselves into flowering Abhyas, cannot perceive photoperiods. Sugar beet, cabbage and carrots are monocarpic plants. To initiate flowering, LDP must be exposed to light for a period less than critical duration. Epton causes thinning of cotton, cherry and walnut. So we have to find out among this which is the false one. Okay. So now we have to know that the concept here, when you look into the, we'll go from here. You see this epithon, okay, it is something which causes thinning of cotton, cherry and walnut they have given. So here epithon is something that is very much which causes the ripening in tomatoes, apples. It is something that is going to accelerate what is called abscission. What is abscission? It's premature fall of fruits, leaves and all that. So the thinning of cotton, cherry and walnut, this is true regarding it. Okay, next coming to that sugar beet, cabbage and carrots. Okay, so they are all called the biennial monocarpic plants. Okay, so they flower and they die in the second season. So that is why they flower, they die in the next season. So they are called the monocarpic plants. So this is true. What about the shoot Abhyas? The shoot Abhyas, okay, sometimes they modify themselves into that flowering Abhyas because they cannot perceive the photoperiodism. So that is the reason that this is also the right statement. But here you see they have given here to initiate flowering, the long day plants must be exposed to a light, okay, that is less than the critical duration. No, they are long. So what it should be, that is a plant that flowers only after getting exposed to light period longer than the certain critical length. So you have a certain period. So we have what is called the critical duration. It should be more than the critical duration. Then only, like in summer or not. So then only that can initiate the flowering. So it should exceed the critical duration. The best example we can give is the spinach, lettuce and some wheat. They are all long day plants. So according to our question, which of the following statement is incorrect? Yes, this is the incorrect because it should be more than the critical duration. So this, all these are all the right one. That means it's wrong, but this is the wrong one, which is the right statement. Okay. So that is the right statement here. Yes, students, please do participate. I wanted you to give the answers. So here we will see the next interesting question that is popping out from the physiology, human physiology. Okay. So when a neuron is in resting state, okay, that is not conducting any impulse, the axonal membrane is comparatively more permeable to sodium ions and nearly impermeable to potassium ions, equally permeable to both sodium and potassium ions, impermeable to both sodium and potassium ions, comparatively more permeable to potassium ions and nearly impermeable to sodium ions. So I always wanted my students to understand that our cells, when they are in the resting state, the inside is negatively charged and the charge that is surrounding that cell is positively charged. Okay, that we call it in a resting state, right? When a threshold is applied, when a threshold stimulus is applied, there is a reversal of these charges or reversal of the polarity, which we call it as the development of the depolarization. That is, there is entry of the positive ions into the cell, okay, and so that is bringing of more of the positivity inside the cell. We call that as depolarization. After a certain amount of period, what happens? This there is going to be reversal of this. That is, the positive charges now they will start moving out of the cell and they will return back to their original resting state. Okay, they will become negative now. That we call it as the repolarization process, which is a part of the action potential. So when I say nerve impulse, it is actually a depolarization, repolarization, that is an action potential. Now here they are asking a question in the resting state. How is this axonal membrane? Definitely what is going to be there? They will not allow the sodium to enter. Rather, it is going to be, what is happening? There is going to be permeable to potassium ions and nearly impermeable to sodium ion. See, a neuron during the resting state, that is when not conducting any impulse, the axonal membrane is comparatively will be more permeable to potassium and it is nearly impermeable to sodium. So that is very, very important. Then what about the resting membrane potential? The resting membrane potential, which I explained, no, that they will be in the resting state. It is around 70 millivolts. Suppose imagine this is the neuron. Acha. Okay, you are putting a CRO. You are putting one electrode inside, another electrode here. Okay, and you are connecting to a cathode ray oscilloscope and you are finding what is the potential difference. So it is around in a neuron minus 70 millivolt. That is, the inside is 70 millivolt less than the outside. So at rest what will happen? The sodium will be more outside and there will be more of potassium inside the cell. But once when a stimulus is applied, there is reversal of this polarity, that more sodium will start to enter inside the cell. So this is the basic concept. Yes, very good. Tasmim, you have given the right answer. Yes, very good. Yeah. Now in gymnosperms like Spinus and Cycas, the endosperm is triploid, haploid, diploid or tetraploid. Now in gymnosperms, like we know the Cycas, Spinus and all there, the endosperms of the female gametophytes, they will be haploid. Only when you see in angiosperm, right, that endosperm where what will happen, it will have that triploid. That is probably the triploid will be there in angiosperms, probably that develops after the fertilization. So see, in gymnosperms like Cycas and Pinus, those endosperms, what they have, they are haploid. Okay, so they are formed before the fertilization. But what in angiosperms, we have that double fertilization, right? So that will be, endosperm is actually triploid, that develops after the fertilization. Yes, very good. Now we will see about which of the following 2 out of the 4 statements are incorrect. The first, transgenic buffalo Rosé produced milk which was human alpha lactalbumin enriched. Restriction enzymes are used in isolation of DNA from other macromolecules. The third option, downstream processing is one of the steps of rDNA technology. Disarmed pathogen vectors are also used in the transfer of rDNA into the host. So they are asking, like, among this, which of the 2, so they have given 4 statements. In that, out of that 4 statements, 2 are incorrect. So they just wanted us to find out the answer. Now first we will talk about this Rosé. This Rosé is the first transgenic cow. Yeah, its milk was very rich with that human alpha lactalbumin enriched. Okay, so now you have to know that it's actually a cow. Okay, so you the other one, the restriction enzyme that is used, what do you call, that is used in the isolation of the DNA, probably by cutting the DNA from the other macromolecule. So which are the 2 we are going to find? The downstreaming is one of the step of the rDNA in the technology. So it involves the extraction actually. It is one of the steps that is involving the extraction and purification. Okay. So what about the disarmed pathogen? The disarmed pathogen vectors are also used in the transfer. So like agrobacterium, tumefacens, like that we will be using. So which of the 2 statement we can take here? That is that statement 1 and statement 2. So they are the right answer here. Okay. So now coming to the next question here. Okay, the next question here, refer the following diagram and identify the labeled parts of kidney. So when you look into the labeled parts of the kidney, okay, so you can see here what are the parts A, B, C, D and E. So what is the first one? The first one A is actually referring to the, what is A referring to? The A is referring to the nephron. The A is referring to the nephron. The A is referring to the nephron. Okay, those structures. And what is this B? It is a cortex. C is medulla. D is the renal pelvis. And the renal pelvis will continue as the ureter. So this is the renal pelvis, and renal pelvis will continue down as the ureter. So what should be the right answer here? It is the option. So nephron, cortex, medulla, renal pelvis and ureter. So this is the right answer. Yes, very good. Which of the following is a naturally occurring growth inhibitor? IAA, ABA, NAA, GA. So what is the naturally occurring growth? Naturally occurring, that because some are synthetic products are also given. So naturally occurring growth inhibitor, the one is what? The ABA. What is that ABA? It is the Aspic Acid. Now this Aspic Acid is a plant growth hormone that is probably important for inhibiting the growth. It is the main one that is going to inhibit the growth. It is a natural growth inhibitor that is produced in the plants also. So this hormone is going to cause inhibition. Inhibition of what? The growth of root as well as the shoot. It also causes abscission. I said no, the premature fall of fruits, flowers, leaves and all that. So this hormone also suppresses the other process. That's why we say that stressor, that has got that anti-stressor effect. So Aspic Acid. So Aspic Acid is a growth inhibitor and it is produced in plants during abscission. Whereas the indolestic acid or naphthalinic acid and GA, they are all of, these are two are the synthetic forms of auxins. Okay, so this is a natural one. So they are all growth promoters. So the right answer would be what? The Aspic Acid. Okay. So next is gel electrophoresis. Gel electrophoresis is used for cutting of DNA into fragments, separation of DNA fragments according to their size, construction of recombinant DNA by joining with cloning vectors, isolation of DNA molecule. So first you should understand that gel electrophoresis is a technique where we are going to separate the DNA fragments. So we are going to separate the DNA fragments. So how are we going to separate? We all know that DNA is negatively charged, okay, and when we are going to put it in a tank and it is connected to that electric, when you are passing through, the DNA being the negatively charged will move towards the positive electrode. So larger the size of the fragment, less will be the separation. So gel electrophoresis is a size, is a technique where we are going to use the separation of the DNA fragments according to their size. So the option is this. So you are going to put it like this, which we have that agar-agar gel and we will have that plate here. Okay, so what happens, we will have what are called lanes in Sulu. So we will be putting the sample, whatever the desired DNA sample, and we will be connecting it to the electrode. We already know that DNA being negative, it will move towards the positive pole. Okay, so larger the size of the fragment, lesser is the separation and vice versa. So here gel electrophoresis is not only used to separate DNA. So it is something that is also used to separate macromolecules like DNA, RNA, proteins, like that we will use. Okay, and the DNA fragments, they will be separated here according to the size. So proteins can also be separated according to their size and charge. So gel electrophoresis we are using for a very vast range of things. Where all we are using this in our forensic purposes, we are using particularly for paternity dispute, even DNA fingerprinting, all these places we are using. Even to check a PCR reaction also we are using the gel electrophoresis. Yes, very good students. I see you all participating, giving right answers. Excellent. An ecosystem which can be easily damaged but can recover after some time if damaging effect stops will be having option A, low stability and high resilience, high stability, low resilience, low stability and low resilience, high stability and high resilience. So when you talk about in ecology, what is that resilience they are talking about? It is the capacity of the ecosystem to give response. So it is a response that is given by that particular ecosystem following a damage, and how fast they are recovering quickly out of it. So that ecological stability is actually determined. It's a resistance property of the community to remain unchanged when subject to disturbance. So that you need to know. So an ecosystem which is having such low stability, that can be easily damaged. So an ecosystem should have a high resilience, will take less time to recover. So what is that resilience? The capacity of the ecosystem to recover, that is to give response to a disturbance by damage and recovering quickly. So an ecosystem, how it can come back after that, it should have low stability but high resilience. So that is the right option. Okay. Yes, excellent students. In bioluminescence, the stored energy changes into light energy, radiant energy, chemical energy and mechanical energy. So bioluminescence is a process, that is emission of light from the organism, where you are seeing the chemical energy is getting converted into the light energy there. So what is happening? There is one organism that is going to produce that luciferin, that is the substance. This luciferin is the product, and that enzyme is called the luciferase. Okay, that will react with oxygen and that will produce the light actually. So bioluminescence is the one, is the emission of light by the organism, like they will glow during the night, that glow of a bacteria, or you can see the flickering signals of the firefly and all, no, they are all characteristic like that. So what is for that? There the chemical energy is getting converted here into the light energy. Okay. So during isolation, yes, very good students, you are giving the right answer. During isolation of the DNA, addition of which of the following causes precipitation of purified DNA? So when we are isolating the DNA, actually we saw that phoresis, right, in gel electrophoresis. After that, once when they move to their respective poles, now it's a time for us to select, like separate it out. So how is that being done? So during isolation, what will be used? Chilled ethanol, ribonuclease enzyme, DNA polymerase and proteases. So first you need to understand that DNA is obtained by treating, sometimes we will obtain it by treating it with some bacterial cell or some plant cell with enzymes like lysozyme or cellulase if it is a plant cell. If it is a fungi, we will use chitinase and all. Now suppose if we are going to use this purified DNA, finally when we want to precipitate, we will use chilled alcohol, that is chilled ethanol. Then only we can get that fine thread of the DNA. So you can see that DNA is obtained by treating the bacterial cell, plants or plant tissue with enzyme like lysozyme if it is bacterial, cellulase if it is a plant cell, chitinase if it is a fungus. So as long as we know that the DNA is intertwined with proteins like histone, the RNA you can remove it by treating with ribonuclease. How can you remove the proteins from that? By treating with protease. So everything will get removed. Now it's exactly that you are going to remove the DNA. So you are going to precipitate by adding the chilled ethanol so that you can get the fine thread of it. So actually they say that the collection of fine threads in that suspension, you can easily collect, what we call it as DNA spooling, we say. DNA spooling we say. Okay. So that is how we do this technique. Yes. Yes, very good. Protestant genome has nucleoprotein in direct contact with cell substance, gene containing nucleoproteins condensed together in loose mass, free nucleic acid aggregates, membrane bound nucleoproteins embedded in cytoplasm. So first you should know here that protisteans, okay, they are eukaryotic organisms. Okay, so they have well defined membrane bound nucleus that will enclose the genetic material and all the organs in the cytoplasm. So here the protisteans are eukaryotes that have true material, the nuclear material is getting embedded. So the DNA is not just like that seen naked, it is going to be associated with the histone protein. So that is with the protista. So you see the membrane bound nucleoproteins that is getting embedded in the cytoplasm. So all the other options are wrong. Okay, so that is the right answer. So which of the following ecological pyramids can be inverted? So what are the ecological pyramids that is inverted? Pyramid of energy, pyramid of number, pyramid of biomass. So among this, like whether it is only A and B, the other option is only B, third option is only B and C, or all the three. So what you have to know, we know this answer. It is a very direct question. Here the pyramid of energy is always upright, whereas the pyramid of number or the pyramid of biomass, they can be inverted. So you can see what is the option here, like only B and C, they can be inverted. Why the pyramid of energy is always upright? Because the flow of energy is always unidirectional. It is unidirectional, that is from the sun to all the, that is to the producers and to all the consumer levels. That is why it's upright. Whereas when you see the energy, it decreases at each trophic level, as when you pass from one trophic level to the other trophic level because of various activity. So there will be 10% loss of all these energy in the trophic level, which we call it as the 10% law. 10% loss of that energy, which was proposed by Lindman. Okay, so that unit, so that energy which is lost by each trophic level, which we call the 10% loss of that energy. So the pyramid of biomass, when you see, it is actually a graphical representation of that biomass which is present per unit at various trophic level. You can see the grassland, trees, those ecosystem and all, they'll be upright. Okay, so the biomass of plants that produces, they'll also be like that only. So suppose when there is any any instability or something, the pyramid of biomass or the pyramid of number, they can be inverted. But always the pyramid of energy, because of the unidirectional flow of energy from the sun to the various trophic levels, is always going to be like in unidirectional from sunlight to the producers to the consumer level. The exact thing, the pyramid of energy is upright. Then we will go to the next interesting question here, where we have during oxygen transport, the oxyhemoglobin at the tissue level liberates oxygen to the cells because oxygen concentration is high and carbon dioxide is low. Okay, so that is one they have said. Or the oxygen concentration is low, okay, and the carbon dioxide is high, oxygen tension is low and carbon dioxide tension is high. Or it can be oxygen tension is high and carbon dioxide tension is low. Now you need to understand here one concept here, because of, see how much is the partial pressure of oxygen in the oxygenated blood, it is 95 mm of Hg. What about in the tissues? It's 40. Carbon dioxide is 40 and in the tissues it is 45 mm of Hg. When there is going to be low oxygen tension and more carbon tension, what is going to happen? There is unbinding of oxygen with hemoglobin and oxygen is released to the tissue. This oxyhemoglobin, after reaching the tissue, there is unbinding and it will be released. Now once in the alveoli, what is happening? Because of that high PO2, there is binding of oxygen with the hemoglobin, okay, and that is going to diffuse into the tissue. Now what should be here? Oxygen tension is low, so they are asking during oxygen transport, the oxyhemoglobin at the tissue level liberates oxygen to the cells. So how does it gives? It has got a very strong bonding here. How does this oxygen is liberated to the tissue? Probably because of the low oxygen tension and the carbon dioxide tension is more. We have what is called the Bohr effect. I hope if you remember that. So in Bohr effect, what has happened? There is increased carbon dioxide. It is going to unbind the, or it is going to prevent the binding of oxygen and with that of the hemoglobin. So that should be the answer. Okay, so you are able to follow? Yes, very good students, you are giving the right answer. Excellent. In rainy season, the door gets swelled up, right, due to imbibition, diffusion, transpiration, respiration. So what should be the answer? So the answer is what? In rainy season, we all know, yes, we can't close the doors, those woody doors. Probably the doors get swelled up because of the phenomenon of imbibition. So we have here what is happening? You don't have actually what you call substance there. It is just that fluid is getting absorbed. So that absorbent, whatever that fluid is taking up without forming a solution, We call that as the imbibition. Like not only this, even when you soak the seeds in the water, no, so what is happening there, that overnight soaking and all, probably that is because of the imbibition. So that is what happens in the rainy season, where the door gets swelled up. Whereas the diffusion is the movement of molecules from region of higher concentration to lower concentration. Transpiration is that of the water loss. Respiration is another different phenomenon with exchange of gases. So imbibition is the right answer. Okay. The function of companion cell is to provide energy to sieve cell, okay, sieve elements for active transport, provide water for phloem, and load sucrose into sieve elements by passive transport, load sucrose into sieve elements by active transport. So here see companion cells, they are going to move the sugar and amino acids into and out of the sieve elements. Like we know the components of phloem, right? So they have those companion cells, they have those sieve elements. So what is going to happen? The companion cells are the one which is going to push that sugar, the amino acids from into and out of the sieve elements. So you will have two areas. One area is called a source, another area is going to act like a sink. So in the source tissue like leaf, the companion cell will use this transmembrane proteins that is going to take the sugar and the amino acids by active transport. Now there is going to be movement of the sugar in the phloem, okay, from the source, where the sugars will be actively, they will enter into the sieve tube. So there is going to be loading, setting up of this water potential gradient. So there will be movement of sugar. So how is that sugar loading happening in the sieve elements? It is by active transport, not passive transport, nor water or provide energy and all. So the exact answer is from the companion cell, that is from the source to the sieve elements, how it is getting loaded up through the active transport. So that should be the right answer. Okay. So now we have another question here. A plant leaf is found to have bundle sheath cell. So when I say this, imagine immediately go for C4 plants. So that is how you need to connect and answer the biology questions. So bundle sheath, C4. Having large size chloroplast full of starch granules. So bundle sheath. Which of the following characteristic can be observed in this plant? Stomata open at night. Okay, so this option is cannot be. So what is happening there in the C4 plants? So in C4 plants, when you look into it, they have the characteristic Kranz anatomy. So in Kranz anatomy, you have two type of cells, the bundle sheath cells, and then you have the mesophyll cells. So here we have what is called in C4 plants, the carbon fixation is going to take place twice. First it is going to take place in the mesophyll cells, then with the help of the PEP carboxylase, okay, and then what is happening, second it is going to take place in the bundle sheath cell in the presence of rubisco. So these are all the correct statements. So what should be your answer? Yes, so what are the right characteristics we are seeing? Yes, presence of PEP carboxylase in mesophils, presence of rubisco in the bundle sheath cell, and what is the light reaction and carbon fixation will occur in different cell types. So that is going to happen. So because bundle sheath cell has all enzymes for all, but what are the other wrong ones? Stomata open at night? No. High photorespiration in hot? No, that is not the right answer. And carbon acceleration rate will be saturated early in the morning? No. So the right answer pertaining with the C4 plants is this is the right characteristic. Okay, so that is the right answer. Yes, excellent students, you are giving the right answer. Very good. Which of the following is not caused by the deficiency of minerals? Chlorosis, etiolation, shortening of internodes, and necrosis. So you should know that which is not because of deficiency. When I say chlorosis, that yellowing, shortening of internodes, necrosis is cell death. So which is not? It is the etiolation. So what is etiolation? The etiolation, when you look into it, it is a symptom that is going to get developed when plants are grown in the dark. Okay, so here example, like you can see pale yellow or white color, that is all because of lack of the chlorophyll. They will have long internodes, okay, poor development of significant tissue. We call that as etiolation. See, necrosis, chlorosis, and shortening of internodes, they are important deficiency symptom, mainly because of deficiency of major elements in plants. But etiolation is because there is absence of sunlight. Sunlight is very much needed for the formation of chlorophyll. Okay, so for the chlorophyll biosynthesis, so the plant will be very pale when you compare with the usual plant. So that is the right answer. So there will be elongation of the internode, the leaves will be very rudimentary. So that is the right one. Okay, so the answer is what? Etiolation. We'll go for the next one. The genetic dwarfness in plants can be resolved by options given are low ABA contents, higher endogenous auxin contents, higher endogenous gibberellin contents, high ethylene content. So when you look into this concept, they say that the gibberellic acid is there, no? When you are going to pour the gibberellic acids on the dwarf plants, we see that what is going to happen? There is going to overcome the dwarfness in plants. Particularly, it's a phytohormone that is responsible for what is called the cell elongation, that bolting and so on. Okay, so that is where with the gibberellic acid. So one of the most important striking effect with gibberellin is elongation of the genetic dwarf varieties, particularly of corn plants, pea plants and all. So it is believed that dwarfism is a mutant variety of plant, that is because of blocking of the capacity for normal gibberellin production. So but when you are going to apply that gibberellin to that single gene dwarf mutant, they will grow to the normal height. Actually, gibberellin is something that will try to overcome the dwarfness in plants. So that should be the right answer. Okay. Yes, very good students. Now coming here for the next beautiful skull, we are going to find out like what is this right things being marked here. So usually we say that the facial bones and the skull bones. So here we are going to see they have given select the option with the correct identification of the structures labeled by alphabets A to F in the given diagram of the human skull. Actually A, they have given it as temporal bone, okay, and this is the frontal bone. Actually the A one, this should be the parietal bone, this is the temporal bone children, and this is occipital. Okay, so B is temporal bone is right. Okay, so this part is right. What about the C? C is the hyoid bone, and E, mandible, is also right. What about this D? This D1 is not, they have given as zygomatic bone. Yes, D is zygomatic bone, but all the three should be correct. So C in this option, A is temporal, A is not temporal, B is parietal, yeah, B is not parietal, so this option cannot be there. C is occipital condyle, this is occipital condyle. Okay, and then what about the D, zygomatic bone, E is mandible, but one is wrong. So the right option here you can see is with the C. So C, you can see here that this part is the, what is this part? It is the occipital condyle. This is the hyoid bone. D is the zygomatic bone. So this part we will call, this portion, no, this is the zygomatic bone, this prominence. This is the mandible, and we can put forth your fingers and you can touch and see one protuberance is there, that part, okay, that is occipital protuberance, but this is the occipital condyle. Okay, so you can see that others all they have mismatched, they have not given it rightly, but I will tell you the right one. This is the frontal, the parietal, the temporal, the occipital. Okay, this is a zygomatic, maxilla, mandible, hyoid, occipital condyle. So you can remember like this. But others are, because immediately, you know, when you look into, many children will make the mistake. Just above your ear is temporal mark. Okay, so you think like that, and this part, this is their parietal, and back one is the occipital. That is why if somebody hits, you have this blacking out, occipital, visual path. So occipital. So like that you remember. This is maxilla, mandible. Okay, like that you can remember. Achha. So concavalin A. The concavalin A is alkaloid, chlorophyll, terpenoids, lecithin. So concavalin A is actually a lectin, which is a carbohydrate binding protein, which is extracted from that jack bean, Cannabilia ensiformis, which is a legume lectin family. So concavalin A is what? It's not a chlorophyll nor a terpenoid, like it's not like that secondary, it's a lectin. Okay, you remember like that, that is more enough. Which of the following is pollinated by water? Viola, Yucca, Oxalis and Zostra. So when you look into the transfer of pollen grains, okay, from by the pollen to that stigma of another flower, if water is used as an agency, we call that as hydrophily. So that pollination through water, we call it as hydrophily. Two types of hydrophily is there. One is called hypohydrophily. That means pollination will occur below the water surface, like Valicernia, like Zostra and all. But I'm sorry, Zostra is below. Valicernia will happen on the surface. Okay, so that is called epihydrophily. The pollination will occur on the surface of the water. So the best example is Zostra, which is hypohydrophilous flower. Achha? So you can see here the transfer of pollen grains from anther of one flower to the stigma of the another flower when water is used is hydrophily. The hydrophily is of two types. We have hypohydrophily. So see, this is the surface. If it is going to be under the surface, we call it as hypohydrophily. If it is going to be on the surface, it is epihydrophily. So what is going to happen? This Zostra, they will have needle like pollen grains without exine. Okay, so that is there. But what about here? The pollination is on the surface like Valicernia. Okay, so very good students. I see you all giving the right answer. Yes, very good students. So Zostria is the right answer. Which of the following tissue is absent in vascular bundles in monocot stem? Cambium, xylem, phloem and all of these. So they have asked like which of the following is absent in the monocot. So just when you are looking into the picture, you can very well make out it is the cambium. See, in monocots, the xylem and phloem, they are organized in the vascular bundles. They will be scattered throughout the stem. Okay. Now as the plant that is growing, the new vascular bundles, okay, you can see it will start forming a new tissue. So the monocot stem, okay, it will have a very simple arrangement than the dicot. So basically what you see the pith, okay, and you will have the vascular bundle and the pith that surrounds them. So that is the thing you will see. That is monocot stems, they have their vascular bundles on the outer, near the outer edge of the stem. So the bundles are surrounded by that large parenchyma. Okay, so that you can see. So there is no pith region in the monocot. Whereas in dicot you can see that ring, okay, but that ring surrounding the parenchyma cell with the pith. So that is the difference. So the answer is cambium. Yes, very good children. You are giving the right answer. Excellent. In humans, the placenta is derived from yolk sac, amnion, allantois and chorion. So we are very much aware of it. So what is going to happen in human placenta? We have a maternal tissue and then we will have the fetal tissue, right? So they are all derived from the growing embryo only. So chorion is actually embryonic derived. Okay, so the chorion is actually embryonic derived, where you can see here, which is got so much of vascularization. Actually placenta is actually a temporary organ which is going to provide the main support nutrition for the growing fetus from the mother. Okay, so this human placenta will have the chorionic villi. Okay, so that is why it is called the chorionic placenta. Okay, it will contain that blood vessels. See, you can see the blood vessels. They are formed from that allantois, the trophoblast. We have syncytial trophoblast, like trophoblast, that inner mass of cell, no? So that will be organized into the chorionic villi. So the answer will be chorion. Okay. Select the correct option for A, B, C and D, which is given in the figure with respect to the relative contribution of various greenhouse gases to global warming. So they have given lot of options here, the carbon dioxide, CFC, methane and NO2. So what should be the right option? So you can very well see here the right option A, that is the A, the 6% is occupied by N2O, the B is occupied by mostly the what you call the CFC, okay, which is contributing 14%. The next proportion is the methane, okay, contributing 20%, and the last larger proportion, that 60%, is what? It is the carbon dioxide. So the right answer option is this N2O, CFC, methane and DCO2. Yes, Srivatsan, you can leave. Thank you. So you have these options. Okay, so this is the gases that is bringing about that global warming, and other issues pertaining to it, okay, that greenhouse gases. Okay, so this is the right option. Next, which one of the following is the correct match of events occurring during the menstrual cycle? So you know, menstruation is breakdown of myometrium and ovum not fertilized. So they are asking to give the correct match of events. See, when I say menstruation, it is the breakdown of endometrium, not myometrium. So this is not the option. What about the proliferative phase? The proliferative phase, that is rapid regeneration of myometrium and maturation of Graafian follicle. So what is happening here in proliferative phase? In proliferative, that is, see, please, all of you, please remember this. Okay, so 0 to 5 day, see, in a woman 28 days is a cycle, menstrual cycle, 28 day once. Okay, probably we will have from 25 to 35 days normal. So 0 to 5 day, we call it as the bleeding phase. 5 to 14 day is the proliferative phase. Day 14, they are going to have that ovulation. 14 to 28 day, we call it as the secretory phase. These changes are happening in the uterus, we call that as the menstrual cycle. The same changes will also happen cyclically in your ovary, that we call it as the ovarian cycle. Achha. Now, in proliferative phase, there is rapid regeneration of myometrium? No, it is the endometrium. And at the same time, there is maturation of the Graafian follicle. Yes, it's quietly growing up. So, this cannot be the option. There is development of corpus luteum in the secretory phase. Yes. So, what is going to happen? The follicle is going to develop here. On day 14, because of LH, there is going to be ovulation, and there is going to be, after the egg is released, that follicle, we call it as corpus luteum, because it will be filled with that lutein cell, yellow color. So, that is why we call it as corpus luteum, and that is going to secrete progesterone. Okay. So, what is going to happen? It is increase the secretion of progesterone. This is the right one. Ovulation, LH and FSH will attain peak level and sharp fall in the secretion of progesterone. So, this also will not be the answer. So, the secretory phase is also called the luteal phase. The secretory phase is called the luteal phase because we have the luteinizing hormone secreted from the anterior pituitary gland. This LH is the one. We use the word called LH surge in solution. So, that is going to cause the ovulation, and this LH is going to stimulate that follicle to produce the develop into a corpus luteum, and corpus luteum will secrete the large amount of progesterone. Yes, so that should be the answer. So very good, you have given the right answer. Excellent. Yeah. You are given a tissue with its potential for differentiation in artificial culture. Which of the following pairs of hormone would you add to the medium to secure shoots as well as roots? Okay, so they have given you thing. What are you going to add? IAA and gibberellin, auxin and cytokinin, auxin and asbicic acid, gibberellin and asbicic acid. See, auxin and cytokinin is the right answer, because when you look the auxin and cytokinin, okay, they are the phytohormones, which is probably very much important for shoot and root development. So, you have a probable ratio of applying the auxin and cytokinin to the plants. Like cytokinin is something which promotes the cell division in the shoot apical meristem, differentiation in the root apical meristem. Okay, and what about the auxin? The auxin is the one that is regulating the growth. It also causes the apical dominance. So auxins and cytokines are used in the development of root and shoot in a cell culture medium. Okay. Germ pore is a region where exine is thick and uniform, absent. I hope you know the structure of the pollen grain. The pollen grain has got two layers. Okay, it has got the inner intine, which will be very thin, made up of the hemicellulose, cellulose, pectin and all. The outermost part is the exine, which will be very thick. Okay, and it will have a characteristic part in it, which is called the sporopolylinen. These sporopolylinen, like you see the entire structure of the exine, at various intervals they will have a break. Those opening, we call it as the germ pore, the furrows, where the sporopolylinen is absent. Okay, so the germ pore is a region where exine is going to be, okay, it is going to be like a small pore in the outer wall, okay, through which the germ tube exits upon the germination for the germ tube to pass out. So it can be apical or eccentric in its location, and on light microscopy, like you can visualize as a light colored area on the cell wall. So that is going to be there. So germ pore is a region where exine is absent. Okay, so that is there. Pentamerous actinomorphic flowers, okay, bicapillary ovary with oblique septa and fruit capsule or berry are characteristic features of family Liliaceae, Asteraceae, Brassicaceae and Solanaceae. Pentamerous actinomorphic flower means it will have that multiples of 5. That is, a flower can be divided into 2 equal halves in one plane. Okay. So the gynoecium is bicapillary there. They will have syncarpus, that is, they will have the superior bilocular ovary will be there. The locule will be axile placentation. So what is that? The members of the Solanaceae family. So they are the one which will have that obliquely placed septa. That is, the oblique septa is because they have the shifting in the position of the ovary. So the nightshade family, the Solanaceae, that edible plants and toxic plants, both, you can say the edible ones we know, eggplant, potato, tomato, all belong to the Solanaceae. So pentamerous actinomorphic flowers with bicarpellary ovary, syncarpus, forming that superior bilocular ovary. They are all characteristic of Solanaceae. Okay. Yes, very good students, excellent. Out of 64 codons, the 61 codons code for 20 types of amino acids. This characteristic is known as degeneracy of genetic code, overlapping of gene, wobbling of codon, universality of codon. So when I say the genetic code, okay, of 20 essential amino acids, the 61 is going to code for 20 type of amino acids. See, you should understand one concept here. Out of 64 codons, the 61 is coding for 20 type of amino acids. This character of genetic code is what we call it as the degeneracy, the degeneracy of genetic code. The degeneracy of genetic code is the reductancy of the genetic code that is exhibited as the multiplicity of the 3 codon combination which is specifying an amino acid. Okay, so that is very important. So the codons that specify the amino acids, they are called synonym, and these codons usually differ in the last phase of the triplet. So out of the 64 codon, only 3 will signify the stop codon. So there is more than one codon for most of the amino acids, and the genetic code will not be overlapping. So 3 successive nucleotides or base codes for only one amino acid. So wobbling will refer to the third base degeneracy. So that is why it is called the degeneracy of genetic code. How many types of gametes will be produced by a plant having the genotype AA, BB, CC? Now this is when you are talking about AA and CC, they are homozygous. What about this BB? This is one heterozygous. Okay. So the number of gametes will be 2n. So the heterozygosity is what? N is equal to 1. So how many gametes will be formed? 2 the whole power 1. So 2 gametes. So it will be in combination of A, B, C. A, capital B, C, or they can be A, B, capital C. Okay, so possible of forming will be what? The 2 gametes. So it will make only 2 type of gametes. The formula is what? 2 the whole power n. How many gametes? How many? Only the heterozygous. Okay, they will have that, right, in the heterozygous state. These are homozygous. So how many? Only 1 is there. So 2 the whole power 2. So only 2 type of gametes will be formed, which are ABC and ABC. Okay, so the answer is 2. Okay, yes, very good, you are giving the right answer, excellent. So smoking addiction is harmful because it produces the polyaromatic hydrocarbons, okay, which causes the reduction in blood pressure, that is decrease in blood pressure, cancer, enhancement of growth of fetus. So what should be the right answer? So when I say smoking addiction, so that is a very dangerous one. Mostly those polycyclic aromatic hydrocarbons, they are going to cause what is called carcinogenesis, we say. The product like polycyclic aromatic hydrocarbons, we call it as carcinogens. So they are the one which is going to produce cancer. So the answer is cancer, because those polycyclic aromatic hydrocarbons, they are going to go and change or mutate like either they will go affect the proto-oncogene, or they will inhibit the DNA repair mechanism, or they will trigger the apoptotic pathways, like that. So they are the one which will disrupt the usual cell cycle, the cell division that is happening normally, and they will induce and bring about what is called carcinogenesis, and that is how the cancer is getting developed. So there will be gene, there are various genes which will take care. So they are going to disrupt that gene function, and that is how it is going to happen. So the substance which will disrupt, we call that as carcinogen, those agents. So polycyclic aromatic hydrocarbon is one such carcinogen. So that is how cancer is coming up. Okay. The percentage of all gymnosperm facing extinction is 12, 23, 32 and 31. So what should be your answer? So here you have to know that according to our IUCN, we have that red list guidelines. Okay. So 12% of birds, 31% of the gymnosperms, and we have around 32% of amphibian species, 23 of mammalian. So what should be the right one? It is the 31. So the answer is 31. So what is happening? More than 15,500 species worldwide are facing the threat of extinction. Presently only 12% of all bird species, 23% of all mammal species, and what we have, the 32% of all amphibian species, 31% of all the gymnosperm species in the world, they face the threat of extinction. Okay. Yes, very good, Tasmeen, you have given the right answer. Interspecific hybridization is the mating of animals within the same breed without having common ancestors, two different related species, superior males and females of different breeds, more closely related individuals within the same breed for 4 to 6 generations. So what is interspecific hybridization? It is one form of what it is, our method of outbreeding. We have outcrossing, crossbreeding, interspecific hybridization. So it is going to be between two different related species. So interspecific hybrids is actually the mating of individual of two different related species. Say a lioness is crosses with a male leopard, the result is a leopon, like that. So here the cross will be made here between the superior male and female of the different breeds, and that is how we will have the what we call it as the interspecific hybridization. So animals within same breed without having common ancestors will be part of outbreeding. Okay, more closely related within same breed is inbreeding. But if you are going to mate it between two related different species, like that will be the interspecific hybridization. But they will be sterile actually, because that will prevent the gene flow between the species. Okay. Variety of cowpea resistant to black bacterial blight is Pusa swarnam, Pusa subra, Pusa sadabdar, Pusa komal. So when you look into this, which among that? So the right answer, when you look through the brassica, it is the Pusa swarnam against the white rust. What about cauliflower? Pusa subra against that black rot. Chili, it is with the Pusa sadabdar, chili mosaic virus. What about with the cowpea? Pusa komal and the bacterial blight. So the right answer is Pusa komal. Which ion is going to help in opening and closing of stomata? Manganese, magnesium, calcium and potassium. So it's a very direct question. If you know that Levitt's theory, we can very well speak about that potassium. Yes, according to the theory of Levitt, that opening and closing of stomata is because of that inflow and outflow, or we can use the right word, influx or efflux of potassium in the guard cell. So the source of potassium, they are nearby subsidiary and epidermal cell. So the stomatal closure is brought about by potassium and chloride from guard cell into the epidermal cell. So that is how they say. So opening, for example, if you see huge amounts of potassium ions will accumulate in the guard cell, that will increase the solute potential. So the water potential will be dropped. So this will draw the water, that is, this will cause the water from the neighboring cell to enter in the guard cell. Okay. So they will become turgid or swollen. So that is how the stomatal pore will get opened, like that. So the reverse will open in case of closing. So the right answer is what? Potassium. Tobacco mosaic virus genes are double-stranded RNA, single-stranded RNA, polyribonucleotides, proteinaceous. So when you take the TMV, tobacco mosaic virus, okay, is a ribovirus that will contain the single-stranded RNA. So all the viruses are nucleoprotein in structure. They have either DNA or RNA as the genetic material. So in a particular virus, either DNA or RNA is the genetic material. Never both are present in the virus. So hence virus can be either double-stranded DNA, that is dsDNA, that like hepatitis B, or they can be single-stranded DNA, which is ssDNA like coliphage, or they can be single-stranded RNA, ssRNA, tobacco mosaic virus. So even retrovirus, no, they have 2 copies of single-stranded RNA like that. Yes, that is the right answer. Yes, very good, Tasmim, you have given the right answer. Which of the following is not the application of PCR? Detection of very low concentration of bacterial virus, detection of mutations in gene in suspected cancer patients, application of the desired DNA segment, detection of antibodies synthesized against pathogens. So when you look into the PCR, the polymerase chain reaction, it is a technique that we are using for amplification of the sample DNA molecule. By using PCR, you can make several hundred copies, right? So it's an efficient, cost-effective way to copy or amplify the small segments of DNA. So what we are doing in COVID now, we are taking the swab test, we are putting it using the lysis buffer, putting in the RTR machine so that we can obtain the DNA sample. So you cannot use for detecting the antibodies synthesized against the pathogen. So that is what is not the application, whereas you can detect even the low concentration of virus or bacteria. That is what we are using in the COVID test now. Or you can detect if there is a mutation in the gene of the suspected cancer patient. You can amplify whatever the DNA segment you want, even 100 copies of it. But antibody detection cannot be made. Okay. Okay, ELISA is that is based on the antigen antibody. This is PCR. Okay. So pneumatic bones are expected to be found in pigeon, house lizard, frog, tadpole, flying fish. So you can see the pneumatic bones. Okay, the bones of aves, particularly the birds, they have those pneumatic because they should be weightless. Then only the bones will contain those air cavities that will reduce their body weight, so that can help them in flying. So the pneumatic bones are found in birds. They will be hollow bones, and because of that, that is making them to fly. So even you can say that what you call the birds, okay, that skull, humerus, all these bones, you know, even the lumbar, they all will be just filled with air cavities. So that will make it very lighter. You can see here, it will be making it very lighter so that they can easily fly in the air. Yes, pigeon is the right answer. The tendency of the body to manifest a characteristic and unpleasant withdrawal syndrome if a regular dose of drug or alcohol is abruptly discontinued, it is called habituation, dependence, psychotherapy, tolerance. So dependence is actually the psychological state that is there in a drug user, okay, in which usual or increasing dose of drug is required to prevent the onset of withdrawal syndrome. So withdrawal symptom will be like anxiety, shakiness will be there. Sometimes the withdrawal symptoms will be very severe. It can be life threatening, like where they will go for suicide attempt and all. So it will lead to ignore them from the social, where they will go in for certain what you call entering into some antisocial activities like that and all. So that is where we say that dependence is tendency which is manifesting, which is a characteristic or some unpleasant withdrawal syndrome if regular dose is abruptly discontinued. So coming to the last question, which among the following can be seen in the ultrastructure of both eukaryote and prokaryote cells? Ribosomes, nucleoproteins, chloroplast and leucoplast. So what should be the right answer? So you can see the eukaryotes and prokaryotes, okay. All these cells, okay, when you will be having what is called the ribosomes, the cytoplasm, DNA and all. But you can see that double membrane, that is the one which differentiates from them. But you can see the what you call in the eukaryotes and prokaryotes, the ribosomes are the one which they are the small electron microscopic viewed structures. They will be actually floating in the cytoplasm. Okay, so where you can see both in eukaryotes and prokaryotes. Okay, so the presence of this ribosome is something that you can view it using the electron microscope. Okay, So that is the main difference, whereas other cells is very characteristic, like where you see only in the eukaryote. Okay, so the ribosome is the right answer. So I think students, like we have discussed the next 45 questions of the part 2 in mock 4 session. I hope this would have been very informative, interactive, as well as an interesting session I had. So thank you students, and winding up, meet you all soon in the next detailed biology solution discussion with various questions and answers. Okay, so thank you students.